Good morning, students. Let us start uh, cell chapter. So, in biochemistry, you this chapter you already studied in your 12th standard or before. So, let us go through again as an introduction. It is a brief account of cell. Okay. So, cell basic structural and functional unit of living organisms. That is the basic unit. So cell aggregate together to form tissue. Tissue again together aggregate to form organ. So altogether organs, there are many organs in our body that constitute for system of organisms. So cells, tissues, finally organs form system of organisms. Now you know the importance of cell. Cell is the basic unit, basic functional unit of life. So life of an organism is outcome of all coordinated function of constituted cell. If cell death is there, necrosis, so that affect to the organs even. So everything there is a abnormal things occur. So it is very important cell as a basic unit of life. Okay. So also cell carry hereditary material, what we call DNA. Okay, deoxyribonucleic acid carry hereditary material. Okay, so you should know what is the difference between prokaryotic cell and eukaryotic cell. Prokaryotic, it is the primitive cell. So this cell you can see in blue green algae, okay, and mainly in bacteria. So this cell, prokaryotic cell, you can see in bacteria, and we also call it as primitive cell. So why? Because prokaryotic cell have no definite nucleus. Also, nucleus is not enclosed by nuclear membrane. So you should know the difference between prokaryotic cell and eukaryotic cell. Prokaryotic cell is primitive cell without definite nucleus and is not enclosed by nuclear membrane, and also they do not contain any membrane bound subcellular organelles. Okay, just it is embedded, that is it. So there is no membrane bound subcellular organelle. But eukaryotic cell have definite nucleus. Higher organism, mammals and others, okay, they have definite nucleus, and nucleus is enclosed by nuclear membrane. There is definite nuclear membrane. In further slide you will see cell membrane or plasma membrane, then you will realize cell structure of this one uh, realize the cell structure, okay, of eukaryotic cell. So they have definite nucleus. Nucleus is enclosed by nuclear membrane. Also they contain membrane bound subcellular organelles. What is the meaning of subcellular organelles? In the cytoplasm, whatever organelles present like ribosomes, mitochondria, rough and smooth endoplasmic reticulum, all those things are subcellular organelles which are suspended in plasma, okay, which are suspended in plasma. So animal kingdom, higher organisms are example for eukaryotic cell. So what is the example for prokaryotic cell? Bacteria. So there are cell bond. So what we are studying is here is prokaryotic or eukaryotic cell, higher organisms. So you can see all cell organelles which are membrane bound. So there is definite nucleus. Okay. So but in prokaryotic cell there is no proper nuclear membrane. So in eukaryotic cell, each cell is bounded with the membrane, that is cell membrane. We also call it as plasma membrane. So nucleus is present in the center of the cell. So there are two terms you know already, I think. Protoplasm, cytoplasm, nucleus we call. See, without the nucleus it is cytoplasm. With the nucleus, cytoplasm plus nucleus, it is protoplasm. Okay. So what are the subcellular organelles main? Ribosomes, mitochondria, endoplasmic reticulum. There are two types, rough and smooth endoplasmic reticulum, then Golgi apparatus. So side bags we call lysosomes and peroxisomes. Okay. So next slide is just shows a part of the cells. So as I told, there is a plasma membrane also called cell membrane, nucleus which is located almost center of the cell. So without nucleus, whatever embedded particles they are organelles, they are embedded in the they are embedded in cytoplasm. So there are lysosomes, endoplasmic reticulum, rough and smooth Golgi apparatus, a cytosol, some secretory vesicles. All those things are main components of cell. Okay. Now we have to more concentrate on cell membrane or plasma membrane, because there are they perform lot of functions, cell membrane or plasma membrane. So in eukaryotic cell, there is proper cell membrane or plasma membrane. Okay. So what are the functions of cell membrane? So remember, this is one of the important question for you. Cell functions of cell membrane and structure of plasma membrane or cell membrane, maybe 5 marks essay. So cell membrane maintains definite shape and size of the cell. It gives a shape, size, because it is the external boundary covering definite external boundaries of cells and organelles which are embedded, which are enclosed by this cell membrane. So it also allows selected compounds to be transported across the cell membrane. So cell membrane is semi permeable, so it is selectively permeable, because it allows only selected compounds to be transported across the cell membrane. What are the other functions? Protective function. It protects cytoplasm and cell organelles, because it is just like fence. It protects whatever is there inside organs until cell lyse. There is main important one more function is absorptive function. The nutrients are absorbed into the cell through cell membrane. Okay, nutrients is required, no, for cellular function and respiration. So what are the functions of the cell? Gives shape and size, external boundaries of cells, organelles, it maintains the integrity. It also selectively permeable, it protects the internal organelles including cytoplasm, and it also the nutrients are absorbed in and out into the cell through cell membrane. Okay. So next, continuation of function of the cell, that is cell membrane also perform exchange of gases. Carbon dioxide, oxygen, exchange of gases takes place through cell membrane. So excretory function, this is one of the most important function of the cell, that is the waste product are thrown out of the cell through cell membrane. Also isolating organelles, so also isolating organelles uh, from rest of the cytoplasm, allowing cellular processes to occur separately. So cell membrane isolate organelles from rest of the cytoplasm, separates, allowing cellular processes to occur separately. Also the cell membrane a site for biochemical reactions, and cell membrane, it allows the cell to change shape, because cell can change the shape according to the external environment requirement. So it allows the cells to change shape. So overall these are the functions of the cell. So it gives shape and size of the cell, external boundaries of cells and cell organelles, so that it maintains the integrity. So it is selectively permeable, it protect the cytoplasm and cell membrane. So also exchange of gases, absorptive function, then excretory function. It is also site for biochemical reactions, it allows cells to change shape. These are the some of the functions of cell. So cell membrane is selectively permeable, as I told. It selectively permeable, it don't allow all the ions to enter in and out. Okay, so that is required, otherwise there is a problem, either hypertonic, and it may affect the cellular function, proper function in metabolic activities and all. So it should be selectively permeable. Being hydrophobic, membranes are permeable only to lipid soluble substances. So what is the meaning of hydrophobic? What is the meaning of hydrophobic? Hydrophobic means water hating, like lipid. Hydrophilic means water loving. So being hydrophobic against water, membranes are permeable only to lipid soluble substances, because lipids are insoluble in water but soluble in organic solvent. So they are a transport system to permit the movement of polar molecule, water loving molecules. So we will see the structure of cell membrane, which is put forward mainly by Singer and Nicholson. So according to their study, cell membrane is about 5 to 8 nanometer in thickness. Can you imagine? That is why it requires a microscope to see the cell. So what is the nanometer? 5 to 8 nanometer means, so 1 meter is 1000 centimeters. So again, if you see 1 centimeter is 10 millimeter. So 1 millimeter is again 1000 microns. So 1 nanometer is 1 micrometer is 1000 millimeter. So 1 nanometer is 1000 micrometer or microns. 1 nanometer is 1000 microns. So it comes millimeter, centimeter, meter, centimeter, millimeter, microns, then micrometer, then nanometer. So it is so small, 5 to 8 as this thickness. So 5 to 8 nanometer in thick. So most important thing you have to remember here is this membrane is made up of lipid bilayer. So made up of lipid bilayer. So lipids are what are lipids? They are biomolecules. Okay, the heterogeneous group of organic compounds. Okay, insoluble in water but soluble in organic solvent. So all your cholesterol, other things, fatty acid, or lipids. Okay, so they are made up of lipid bilayer, which is of amphipathic lipid molecules. What is the meaning of amphipathic lipid molecules? They are both hydrophilic and hydrophobic. What is the meaning of hydrophilic? Water loving. Hydrophobic is it is insoluble in water. Okay, so that is the structure of cell membrane or plasma membrane, 5 to 8 nanometer in thick. So they are mainly made up of lipid bilayer, which is amphipathic in nature. That you have to mention in your this one notes, or this very important. So membranes also contains proteins and carbohydrates. Anyway, we will see about proteins and carbohydrates next classes, so no need to worry. So this cell membrane is made up of lipid bilayer. Okay, lipid bilayer, and it also contains carbohydrates and proteins. So there are two diagrams. So which one is suitable for you? You can select and write. So one diagram, cell membrane with the labeled diagram, you have to remember for the exam point of view. Okay, it is fluid mosaic model of cell membrane. Okay, fluid mosaic model of cell membrane or plasma membrane is very important from the exam point of view. So membrane structure is currently accepted is fluid mosaic. So according to this, there is a double layer of phospholipid, that is fat. What is the meaning of phospholipid? It is a compound lipid. Usually it contains fatty acid, glycerol, along with this phospholipid contain nitrogen. Okay, so phospholipids, that is the important function of phospholipid. When you study the function of lipid, you will come to know so importance of phospholipid. Okay, so there is a hydrophilic head, phosphate head, which is hydrophilic, is attached to water both inside and outside, cell to cell in a watery environment. Okay, so hydrophilic head is outside, hydrophobic is inside. Okay, so double layered lipid is there, quite fluid with the proteins floating within it. Okay, so double layered lipid with proteins floated within it. Okay, there is a hydrophilic head. Okay, hydrophilic end and hydrophobic head or end, we will call it as head or end. So one is projected outward, another one is projected inward. Hydrophilic is attracted to water both inside and outside the cell in a watery environment. So this is the main structure of this plasma membrane according to fluid mosaic model. So we go further. So there is one more diagram, so where it is already mentioned about the parts of the cell membrane. We can go through the diagram. This one, this is the okay, simple and easy to remember. There is one more 3D type structure. Uh, so there also it mentioned about phospholipid head and tail. Okay, so head is already projected outwards. There is a phospholipid bilayer, glycoprotein, cholesterol, channel proteins also there. Okay, so cell membrane, we will continue a little bit detail about this cell membrane. Lipid and protein molecules are bound each other by non-covalent bond. Okay, lipid and protein molecules are made up of non-covalent bond. You know, ionic bond, hydrogen bond, covalent bond. Compared to ionic bond, this covalent bond is weak. A covalent bond is formed by sharing of electrons. Ionic is, there is ionic complete transfer of electron, but in covalent bond there is a sharing of electrons. Okay, it is made up of covalent bond. Lipid and protein molecules bound each other by covalent bond. See, this is the important of lipid mainly and proteins because they are part of cell membrane, phospholipids. Okay, so carbohydrates are found lesser amount and are covalently bound to lipid and protein. Lipids are amphipathic in nature. So what is amphipathic? It is both water, hydrophilic and hydrophobic. Phospholipids, they regulate their permeability of cell membrane. They regulate the permeability because transport in and out. It is selectively permeable. So these phospholipids regulate the permeability of cell membrane. Okay. So glycolipids and cholesterol also there, as I told. So glycolipid is nothing but carbohydrate. What are carbohydrates? Your protein, monosaccharide, disaccharide, polysaccharides are carbohydrates. So carbohydrate attached carbohydrate and lipid is glycoprotein, nothing but. Okay, so also cholesterol, it is a sterol. Cholesterol is a sterol compound. Usually carbon is 27, so it contains a cyclopentanoperhydrophenanthrene. It is also derived of lipid cholesterol, important. So why it is important? Cholesterol is required for us. Why required? Hypercholesterol also problem. You heard about the myocardial infarction and other things, heart attack and all. Cholesterol but required. Low cholesterol, why? It is also a part of, main thing is cell membrane. So also these lipid and protein molecules are bound to each other by non-covalent bond. This I finished already. So next is membrane proteins. The peripheral membrane proteins and integral membrane proteins. So there are two main types. Okay, as I told, we are not going in great detail, not required for you. So peripheral membrane proteins are attached to the lipid bilayer on either surface, inner and outer surface of the membrane. So they can be easily removed without disturbing the membrane. Okay, that is the difference between peripheral membrane protein and integral membrane protein. So what is the difference? In case of peripheral membrane protein, they can be easily removed without disturbing the membrane. They are attached to the lipid bilayer on either surface, inner and outer surface. But integral membrane proteins are deeply embedded into the lipid bilayer. So they cannot be easily taken out without disturbing membrane, without breaking the membrane. So in the integral proteins are deeply embedded in lipid bilayer. It is difficult to remove them. Okay. So also carbohydrates are relatively minor components in cell, but they are present as glycolipid what is a glycolipid? As I told, it contains carbohydrate. Glyco means it contains carbohydrate. Carbohydrate plus lipid is glycolipid in simple language. So carbohydrates are present as glycolipid and glycoprotein. Again, what is the meaning of glycoprotein? Protein and this carbohydrate is called glycoprotein. So carbohydrates present as these are both are glycolipid and glycoproteins comes under compound complex carbohydrates or lipids. The carbohydrate coat on the outer surface of the cell is called glycocalyx. Is called as glycocalyx. Okay, is called as glycocalyx. So these are the structure of cell membrane in brain. Okay, so again to repeat, mainly cell membrane or plasma membrane made up of phospholipid bilayer. There is hydrophilic head and hydrophobic end or tail we call. So this is fluid layer. So protein is floated in the double lipid layer. Okay. So glycoproteins, glycolipids and cholesterol also integral part of the cell. So lipids and proteins are bound to each other by non-covalent bond. Carbohydrates present in less amount, that is glycoprotein and glycolipids. So lipid molecules are hydrophilic and hydrophobic. Phospholipid is the main content. They regulate the permeability of cell membrane. So cholesterol also present. So the proteins are two types, mainly membrane peripheral membrane proteins and integral membrane proteins. So peripheral can be easily membrane proteins can be easily removed without disrupting membrane, but it is difficult to remove integral membrane proteins. So what is glycocalyx? Carbohydrate coat on the outer surface of the cell. It is glycocalyx. They are glycolipids and glycoproteins. What are those? They are nothing but they also complex carbohydrate and protein. Okay. Now let us see the cell organelles. Mitochondria, subcellular organelles. What are those subcellular organelles? Endoplasmic reticulum, Golgi apparatus, ribosomes, mitochondria and all. So mitochondria. So in these things you may get a multiple choice question. Which of the following is powerhouse of the cell? So what you have to write? So there will be option A, B, C, D. So mitochondria is powerhouse of the cell. Synthesis of ATP. So what is the shape? Spherical or oval shape? Mitochondria having spherical or oval shape. Okay. They have two membrane. There is outer and inner membrane. See, outer membrane is smooth, while inner membrane is folded to form cristae. So folded inner membrane is it is folded in zigzag manner to form cristae. Okay, that is called CRISTEE. So inner mitochondrial membrane contains the components of electron transport chain. There are two membranes, as I told, outer mitochondrial membrane which is smooth, inner mitochondrial membrane folded to form cristae. Okay, folded to form cristae. So inner mitochondrial membrane contains components of electron transport chain. So it is a separate chapter. So this electron transport chain, what is the thing happens is, It is a cellular respiration, oxidative phosphorylation for the synthesis of ATP. What is ATP? Adenosine triphosphate molecules, which is energy rich molecules, which is required for cell function energy. So that is synthesized in cell electron transport chain, that is cellular respiration, okay, by energy produced by various oxidative reactions. So finally it enters this electron transport chain to form ATP molecules, that is electron transport chain. So inner mitochondrial membrane contains the components of electron transport chain, so that is the importance. So oxidative phosphorylation also, so it takes place in mitochondria, inner mitochondrial membrane. So central cavity is there in mitochondria, it contains mitochondrial DNA. Okay, there are many other reactions which takes place in mitochondria. Okay, so that is the main function, synthesis of ATP, that is the powerhouse of the cell, cellular respiration, that is the important function of mitochondria. Okay, so functions, as I told, inner mitochondrial membrane contains components of ETC. ETC means electron transport chain, oxidative phosphorylation for the synthesis of ATP. Okay, so where the energy is released from oxidation of food molecules is trapped to form ATP. Okay, that is because ATP is synthesized in mitochondria, it is called as powerhouse of the cell. So also beta oxidation of fatty acid, beta oxidation of fatty acid is required for this again synthesis of ATP molecules and energy rich. These are the things comes under metabolism. So some of the students are studying this in second year. So beta oxidation of fatty acid, TCA cycle, tricarboxylic acid cycle, okay, which also comes under carbohydrate metabolism, ketone body metabolism, synthesis of ketone bodies, acetone, acetoacetic acid, and all this also comes under carbohydrate metabolism and lipid metabolism. Ketone bodies, if present, they are abnormal, okay, and in the blood above the level. So beta oxidation, TCA cycle, ketone body formation takes place in mitochondrial matrix. So for many reactions, this is the place, mitochondria or mitochondrial matrix. Okay. So next is the endoplasmic reticulum. Okay, there are two types, as I told, smooth endoplasmic reticulum and rough endoplasmic reticulum. Reticulum means it is a network like, okay, network membrane enclosed spaces that extend throughout the cytoplasm, that is why it gives support, okay, because of the networking type of system present in endoplasmic reticulum. So some of the structure extend from the nuclear envelope to the plasma membrane. They can be easily removed without disrupting membrane. As I told, there are two types of endoplasmic reticulum, smooth and rough endoplasmic reticulum. So what is the main difference between smooth and rough endoplasmic reticulum? Rough endoplasmic reticulum attached with ribosomes, okay, ribosomes, factory for protein synthesis. So rough endoplasmic reticulum, rough in appearance because of the presence of ribosomes, railway tract appearance. Smooth endoplasmic reticulum is smooth, which has no attached ribosomes. Okay, structure I mentioned there. Next, the dot whatever comes is presence of ribosomes in rough endoplasmic reticulum. So there are other structures because it is enlarged structure. Golgi apparatus, suicide bags, lysosomes also you can see. See, they are also attached up to the this nuclear attached up to they extend from nuclear envelope to the plasma membrane. Okay, that is about this one endoplasmic reticulum. This helps in protein synthesis function of endoplasmic reticulum because ribosomes also attached to endoplasmic reticulum. So detoxification of various drugs takes place in the endoplasmic reticulum. Detoxification of various drugs. Toxic means they are poisons. They need to be removed. So that takes place in the endoplasmic reticulum. Synthesis of lipid. Okay, lipid molecules, whatever glycolipids, phospholipids, this TG cholesterol and phospholipid takes place in endoplasmic reticulum. These are the things comes under metabolism. So site of synthesis of cholesterol. So in endoplasmic reticulum. Okay, helps in the protein synthesis, detoxification of various drugs, synthesis of lipid, gives mechanical support to the cell. Because of its network full appearance, it gives mechanical support to the cell. Also important thing is this endoplasmic reticulum serves as a storage of calcium. Calcium is very important compound. Okay, calcium is required for nerve this one as a transport. It is required for many functions, bone and teeth formation, then muscle contraction and all. So it is important, calcium. So this endoplasmic reticulum act as intracellular reservoir of calcium. So Golgi apparatus, next is they are flattened membrane surrounded vesicle. Okay, they appear as vesicle. So Golgi apparatus required for protein modification. Okay, translation protein synthesis is different. After synthesis, for protein modification, protein synthesis takes place in the ribosomes. Protein modification takes place because sorting out of the different proteins. Sorting, packaging and secretion takes place in the Golgi complex. So remember that protein synthesis takes place in the ribosomes. They are even attached to rough endoplasmic reticulum. But what is the function of Golgi apparatus? Protein modification, sorting and packaging to form different forms of protein. Okay. So additional carbohydrates and lipids in the formation of glycoproteins or lipoproteins takes place in Golgi apparatus. Golgi apparatus. So addition of carbohydrate, lipid and formation of glycolipid and lipoproteins. Lipoproteins means lipid and protein is lipoproteins like HDL, high density lipoprotein, LDL, you heard about good cholesterol, bad cholesterol, HDL, LDL, all comes under lipoprotein. Protein plus lipid, they are complex proteins, lipoproteins. Okay, so that is the this one. So after synthesis, addition of protein, addition of carbohydrates and lipid takes place in the takes place. So Golgi apparatus is required for this. Okay, so there takes place addition of carbohydrate and lipid in Golgi apparatus. So I have given this it's their structure, Golgi apparatus. So briefly you should know the structure of Golgi apparatus. So there are other cell organs. Lysosome is also called as suicide bag. So this is also one type of multiple choice question. Which of the following cell organelle is suicide bag? Okay, that is lysosome. So it is a membrane bound vesicle containing various hydrolytic enzymes. Whenever you remember lysosome, you have to remember that it contains hydrolytic breakdown enzymes called hydrolases. So hydrolases present in lysosomes, cathepsin, elastase, DNase, phospholipase. These are the some of the hydrolase enzyme, hydrolytic breakdown which breaks. Okay, so what is the function of lysosome then? Lysosomal enzymes are capable of digesting protein, carbohydrate, lipid and nucleic acid. Hence, they are responsible for degradation and recycling of intracellular biomolecules. Whichever becomes old and degenerated, that is digested, meaning is that is removed from cell by lysosomes. Why? Because of the, because of the presence of enzymes, as I told, all phospholipase, elastase and all. Lysosome also digests substances brought into cell by phagocytosis. Phagocytosis, what is the meaning of that? Cell engulf, cell membrane extend and to form phagosome, so they engulf the external particle. So that is called phagocytosis, cell eating we call. So lysosomes also digest the substances brought into cell by phagocytosis. So I have given brief this one, structure of lysosome, enlarged one. Okay. So nucleus, what about nucleus, which is present in the center of the cell, is the largest subcellular organelle. So double layered nuclear membrane is there because this is the eukaryotic cell. So nuclear pores permit the passage of molecules in and out of the molecules. So if you see the structure, you'll know. So there is a nucleolus inside, nuclear pores are there in between. Okay, nuclear envelope and in between nuclear pores is there. So DNA is the hereditary material present in the nucleus. There is also presence of nucleolus. So what is the main function of nucleus? It carries the genetic material. Okay, carries the genetic material. So nucleoplasm is the ground material of nucleus. It contains DNA rich in enzymes, DNA polymerase, RNA polymerase. Okay, molecular biology will study these things. But it's not there in the syllabus anyway. So this nucleoplasm is the ground material of nucleus. It contains the enzymes, DNA polymerase, RNA polymerase and all. Not only that, there are many other enzymes. Nucleoplasm, particularly in DNA, that is the presence of this nucleus, DNA and RNA polymerase. Nucleoplasm is filled with chromatin. So during cell division, chromatin condenses to form chromosomes. So in the nucleoplasm, there is the presence of chromatin. So cell division, it transforms to condenses to form chromosomes. So nucleus of eukaryotic cell contains dense body which is called as nucleolus, which is rich in rRNA, ribosomal RNA. So nucleolus is rich in ribosomal RNA or rRNA. This is the type of RNA. So functions: DNA is the genetic information it carries to the offspring. It is also required for a synthesis of protein molecule. So ribosomes, again there is a chance of multiple choice question. Ribosome is required for protein, helps in protein synthesis. Nucleoprotein particles present either freely or in cytosol or bound to endoplasmic reticulum. So these ribosomes are nucleoprotein particles present either freely or they are present freely present either freely or in cytosol or bound to endoplasmic reticulum. So these ribosomes helps in protein synthesis. So there are other structures in cytoplasm, that is cytoskeleton. They are non-membranous subcellular cytosolic fractions that possess fibrous structure. So each is a polymer of protein subunit held together by they are held together by weak non-covalent bond. So they are cytoskeleton. They are subcellular cytosolic fraction that possesses a fibrous structure called cytoskeleton. So next, this example for this cytoskeleton is microtubules or microfilament. They maintain the cellular structure shape. Also they required for cell division and motility. Okay. They provide internal framework for positioning of various cell organelles because they are located in the same like shape, fibrous structures. So they provide internal framework for positioning various cell organelles. Okay. So definition for this microtubules and microfilament, non-membranous subcellular cytosolic fraction that possess fibrous structure. So each is a polymer of protein subunit held together by weak non-covalent bond. That is about this cytoskeleton, so which contains mainly microtubules and microfilament. Okay. This is the brief introduction of cell. So main question comes in this chapter is fluid mosaic model of cell membrane. Fluid mosaic model of cell membrane. So that you should remember the structure is important. You have to write the structure. Mainly you can include this. Okay, the structure, and you should mention about the phospholipid bilayer contains other cholesterol, glycolipid, glycoproteins, hydrophilic, hydrophobic, this head and tail. You have to mention about the protein briefly. You have to mention, but diagram is very important. Along with that, there are other multiple choice, as I told. Mitochondria is powerhouse of the cell, and also there is a chance of short note mainly on mitochondria and ribosomes, endoplasmic reticulum. Okay, so these are some of the important question. Also cell membrane or plasma membrane, functions of cell membrane or plasma membrane. So these are the things you have to remember. So remember fluid mosaic model of the cell and functions of the cell membrane is others also important. These are the very important things. Okay, thank you.